let's go back to talking about making proteins. So we had our mRNA sequence. We have our ribosome that with the help of these tRNAs bringing in the amino acid. We're reading the mRNA molecule in these triplet codons. tRNAs are bringing in maybe I'll use this drawing instead. So slightly different mRNA sequence that I had drawn before. So here's AUG with thionine, and then this big shadow here is the ribosome. Then we have the next triplet is GUU, the anticodon complementary. So that's bringing in a valine. This bond is being made. Then as the ribosome moves on to the next triplet, the next amino acid comes in on a tRNA. This bond is made, and then now we can break the bond between the amino acid and the tRNA, leaving the tRNA free, but we still are keeping our growing protein chain attached to the ribosome. So you have to make that next bond before you can break the tRNA to the amino acid bond. And then the ribosome moves all the way across, adding the amino acids in order. And then here's that UGA, which is one of the stop codons. There's a protein called a release factor that recognizes that stop codon and breaks that last bond, releasing the protein chain. So let's look at that actually in
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So hopefully that makes things a little more clear when you can see it happening all at once rather than trying to draw an active process on the board. So there are questions before we go on about the ribosome and the tRNA. Okay, so let's now, we know that the DNA is transcribed into RNA, and then the RNA molecule is translated into protein. So let's take a look at what happens if something goes wrong and there's a change in the DNA sequence, and let's take a look at how that affects the protein that's made. So on the next page of your worksheet, you're asked to transcribe the following piece of DNA to RNA, and then write the RNA sequence, and then translate the RNA molecule into DNA. So let's start. So let's do that first one together, and then we'll take a look at the others when something. You can work in your groups to look what happens when something goes wrong. So if we have TAC, AACT, hopefully that's copied correctly. Did I forget something?
ATCP.
Here at the end?
Yeah.
APC? So let's transcribe this together into RNA. So complementary to T. And let's translate this into protein. So triplet is MET, LEU, leucine, so AAG, lysine, AUC, ILE, glycine, so here's what our DNA sequence should look like, transcribed into RNA and then translated into protein, but there are things happen, there are environmental factors that can cause changes in DNA. So the next part of this asks you to bombard your DNA molecule with X-rays and mutate it so the 6th nucleotide is mutated to a T. So now write the new RNA molecule that's made and the new protein that is made that happens after that 6th nucleotide is mutated and changed from a C to a T.